Good morning. You okay, Mackenzie? Mackenzie's engineering something over there. Okay, so today we're going to pick up where we left off. Nancy's going to teach. No? Okay, good. All right, we're going to pick up where we left off with enzymes. Okay? This is the last part of chapter 6, and then we're going to get into chapter 7. And chapter 7 is going to go real quick because it's cellular respiration. For Wednesday, okay? For Wednesday, there's going to be a little sheet, a cellular respiration worksheet that I want you to print up and bring into class. Okay? I'll post it on Moodle, print it, bring it in. You'll fill that out as we go through cellular respiration. So it's a nice little table of what goes in, what comes out, what the output is, what eventually ends up being established or being produced and consumed, so you can follow it along from point A to point B. All right? But we've been talking about reactions that occur within the cell, all right? And all of those, the sum of all of those reactions that build things up and break things down, is metabolism, okay? And those reactions, we've gone through and described those reactions as exergonic, endergonic, whether or not they happen spontaneously, and that all depends upon the energy status of those reactions. And then we talked about how enzymes help speed up those reactions and help those reactions to go faster based upon lowering the energy of activation of that any particular reaction. Now the next step is, well, how do cells, or how does biology regulate the process of, or the activity of enzymes? All right, because enzyme activity needs to be regulated. Okay? We talked about inhibition of enzymes, right? Whether or not it's a competitive or non-competitive inhibitor. And now we're going to talk about, well, when those enzymes are present and active, when are they shut off, etc. Okay? The first sentence up there is true. If you lack regulation of a protein or of enzymes, it results in chaos. Okay? Lack of regulation of enzymes can lead to cancer. It can lead to cells consuming themselves. It can lead to problems. Things that are unregulated causes lots of problems. All right? So there's a couple different ways that cells do this. You can either regulate the production of an enzyme, whether or not it's made or not, but that tends to be an inefficient process. It takes a lot of energy input to get rid of an old enzyme and make a new one from scratch. Okay? Instead, there are more efficient, easier ways to control activity. All right? So we can either control the expression or we control the activity. All right? The activity is a lot easier to control. All right? And this leads us to what we call allosteric regulation of enzymes. All right? So allosteric regulation can be inhibitory or stimulatory. They can either shut the enzyme off or turn it on. Okay? Or make the enzyme not work as well or make it work better. Okay? The second point up there says that this occurs when a regulatory molecule binds to a protein at one site and affects the protein function at another site. So what do you know about what we covered last class that fits under this allosteric regulation? Two things we covered that actually fit under this allosteric regulation? Sam? A non-competitive inhibitor, why?
Because it's binding to the enzyme, but it's not, and it's affecting the function, but it's not.
So a non-competitive inhibitor binds to the enzyme where? Not at the active site, right? But it affects the enzyme activity at the active site, all right? Or it affects its activity somewhere. So a non-competitive inhibition would fit in this, okay? Cofactors such as vitamins, right, which can interact with enzymes, they will also fit under this, okay? So allosteric regulation is another way, right, competitive or non-competitive inhibition fits under that. All right, so this can be activation and inhibition. These enzymes are made from polypeptide subunits. Each enzyme has active and inactive forms. Okay? Lots of enzymes are produced in an inactive form. And then once they are stimulated through allosteric regulation, okay, they are stimulated and they can be produced or switched into the active form. So in this case, the activator binds to the enzyme and stabilizes the active form, and the inhibitor binds to the enzyme and stimulates or stabilizes, Allie, hello, the inactive form of the enzyme. Right? So allosteric activation stabilizes the active form, inhibition stabilizes the inactive form. Is it binding to the active site? Probably not. Right? Somewhere other one site, but affecting the activity second site. Here are some examples. Okay? So we have allosteric activators and inhibitors. Here's an enzyme with 4 subunits. One of those, one of the areas of this subunit, okay, is an example for, or an area for allosteric regulation. The activator binds and stabilizes this form. Now all 4 of these active sites can bind to substrates, okay? And then here's the inactive form, so it's going to go back. Oscillation means it's going to go back and forth between this. And the inhibitor binds to this site, note the shape recognition, okay, and stabilizes the inactive form. This one's active, this one's inactive. This is allosteric activators and inhibitors, okay? The other example of this, besides non-competitive inhibition, is also something called cooperativity. All right? Cooperation means 2 things working together, more than one thing working together, right? Okay. So in this case, the binding of one substrate to the active site stabilizes the active form in the enzyme and it becomes more active. All right? In this case, the substrate is the activator. Right? So what's going to happen? If this substrate binds and now these other active sites are stabilized, then what happens? More substrate can bind, right? Right? So there's going to cooperation between the substrate and the enzyme that's there. In this case, the substrate itself is the activator. No, wrong one, sorry. Okay, so it can amplify enzyme activity. I'm going to call this a priming. Yes, Keon.
So activators and inhibitors are both regulatory proteins?
Yes.
Okay.
Yes. It's just a matter of what type of regulation that's going to be. Is it going to be stimulatory or inhibitory? All right?
What does it like necessarily mean when an enzyme's stabilized?
Well, if it's stabilized, so what's going to end up, most of the time, there's inactive and active forms, and they're going to flip-flop between the two. They're going to continually, move between one form and the next. But once it's stabilized, it gets stuck there. So if it's stabilized, it gets stuck in that inactive form. Then all of those proteins that are stuck there are no longer, they're not active. So the enzyme activity is going to go down. If it gets stuck in the active form, they're active, and it's going to go. The enzyme activity eventually going to go up.
Okay.
Okay. And then something else has to happen to flip-flop them out of there. Okay. Questions on that? Questions on cooperativity? And then with this allosteric regulation, as a extension of this allosteric regulation, we have what's called feedback inhibition. Okay? Feedback. What's feedback? Not necessarily in this, in biology, or in this. What's feedback in general? All right, so you're going to let me know what's going on. So I ask you questions like this all the time, and I get crickets. That's feedback to me. That lets me know what's going on, right? Either you're not paying attention, sleeping, or you don't know what's going on. If I don't get any answers back, right? Those are the three things that I'm thinking, right? But I ask questions, and you answer, or I ask a clicker question, and I get feedback of, hey, what's the status of the class, right? So feedback happens all the time in cells as well. The cell is constantly taking questions, taking a census of what's going on in the cell, right? Then it utilizes that feedback to adjust, okay? In this case, feedback inhibition, we're going to have a reaction that's going to occur, right? A plus B in the presence of an enzyme, right, is going to yield, product AB, right? Do we want to produce, say, product AB as a cellular protein? Do you want to produce more than we need to? The cell doesn't want to overdo, like excess, because there's no room to store it, number one. Number two, it's inefficient. You're wasting resources, okay? Like, we could use A for something else. We use B for something else rather than producing this, okay? So in this case, what ends up happening is that the production, the product, actually feeds back and regulates the activity of the enzyme, okay? So that not too much product is made. It's a very efficient way of regulating enzyme activity, okay? So now when the product's not there, what happens to the enzyme? Well, the product AB is not there. What does the cell recognize? Does it need it? Yes. Okay? So it needs to make more, right? If there's no AB there, then we need to make more. So if there's no AB there, that means that enzyme is turned on, and now we start to produce more. Okay, we're going to see. We'll go over this picture in a minute. Okay? Let's do it this way. So a lot of these products are also in steps. Okay? So here's an enzyme that produces something that goes into these steps. Okay? Eventually you end up with an end product, which is isoleucine, an amino acid. Okay? Do we want excess amounts of isoleucine in the cell? No, we just want we want the right level that the cell needs, right? Okay, we don't want too much, we don't want too little, right? But these enzymes, excuse me, these enzymes are regulated by the product. They're regulated by the end product here. So here's isoleucine down here, and it feeds back to the initial enzyme, and it's going to inhibit it. Okay? So if I have a lot of isoleucine or a lot of the end product, that's whatever's excess is going to be bound to the enzyme and prevent production of more. Right? Think about, you have flushing toilets, right? You all flush the toilet. Okay, how does the toilet stop? You ever open the back of it and look inside? Right? There's a little float bobber, right? And eventually the water level gets up so high that the float is at the top and it moves and it shuts off the water. Right? That's what's happening. That's a feedback inhibition. Eventually the levels of isoleucine get high enough so it shuts off the enzyme. Raven.
So what is a negative feedback?
This is a negative feedback. In this case, it's negative feedback because we are stopping the activity of the enzyme. Okay? So this is feedback inhibition. Now when we talk about, especially Raven, we talk about negative feedback loops. That's what we talk about in anatomy and physiology about responses to like physiological responses. All right? But this is feedback inhibition. The amount of the end product is going to inhibit the enzyme that's producing it. Okay? What's great about this process for the cell? Does the cell have to make more enzyme? Yes or no? No. It doesn't have to. So this enzyme right here is not consumed. It just is bound right here. It's bound by isoleucine. Allosteric regulation. Carolyn.
What makes isoleucine like unbind?
So it's an enzyme. So now the levels of isoleucine go down. There's an equilibrium of binding and on and off. Okay? So either it's bound or it's not bound, and it's always ongoing. Okay? But as the levels of isoleucine in the cell go down, less of that product is available to bind the enzyme. So now more of the enzyme is not bound and can make more of this until levels go up. Sam.
I'm just a little confused because you said that like the amount of product like inhibits the enzyme, right?
Yes.
But then on the other slide it said that the feedback inhibition prevents cells from wasting chemical resource by producing more.
Yes. So you're going to stop wasting chemical resources. So in other words, I have this threonine right here, right? Threonine right here. That's going to be utilized and changed into isoleucine. But once I get enough isoleucine, I don't want to break down more threonine. Threonine is another amino acid. If I don't stop that process, the cell doesn't stop that process, it's going to waste all of the threonine and just have an excess of isoleucine. So it prevents from wasting chemical resources, the reactants in that process. Okay? Other questions on this? So feedback inhibition is one way of regulating enzyme activity. Okay? So we can regulate enzyme, the amount of an enzyme present. Okay? If there's no enzyme present, it can't work, right? Yes? Second, we can regulate an enzyme's activity through allosteric regulation. And then the third is we can regulate an enzyme's location. All right? We're going to talk about cellular respiration in a little bit and on Wednesday. And that's a three-part process. One part happens in the cytoplasm. Another part happens in the mitochondria. Okay? So if I have a part of this process that I want to regulate that happens in the cytoplasm, I can move the enzymes out of the cytoplasm into a vacuole. I separate things apart. If I separate the enzyme from its substrate, can it work? This is the part where you all together say, no. Can it work? No, good. Why? Haha. Why? Nothing to bind to. They're not interacting, right? So if you separate them apart, they can't interact, right? This is at home when my two daughters are fighting. One goes to her room, the other one goes to the other one's room. They separate apart. I don't have to deal with it, right? Okay? So that's all it is. It's a location. So if I move it from where it's supposed to work, it doesn't work, okay? So you have some enzymes that reside in specific organelles. For example, those that work in cellular respiration are in the mitochondria. You don't find them in lysosomes. You don't find hydrolytic enzymes in the mitochondria and whatnot, all right? So we have a specificity of location specificity for action, okay? Questions on that? Yes, no, maybe. Keep the activity of enzymes and energy flow and metabolism in mind. Because now we're going to move on to the next chapter, which is the process of cellular metabolism. How cells create ATP from organic substrates. Okay? And we already know that energy flows into a system. What's the ultimate source of energy for the biosphere? The sun. Okay? So here is a graph or a infographic, whatever you want to call it. Okay? I think that's working. I don't know. It's recording. There we go. All right. An infographic that shows the movement of energy through a whole ecosystem. It comes in as sunlight, light energy, and what cells harvest that energy? Through what process? Through what process, Mackenzie? Photosynthesis. Why'd you say it with a question mark at the end? Photosynthesis. Confidence, people. Confidence. Okay? Photosynthesis. All right? And then that process transforms the light energy and stores it in a chemical form, in the form of glucose. And then cells have to again, another transformative process, and move the energy from glucose to ATP. And that process is cellular respiration. Okay? So we're going to change the organic molecules into ATP. And then where does energy leave the ecosystem? Eventually in the form of heat. Okay? Otherwise, it gets cycled through. Right? All of this is just a transformation of light energy into usable cellular energy and eventually leaves the system as heat. All right? So, we're going to talk about the catabolic pathways that allow cells to harvest energy. Okay? And this happens, the cellular respiration is what we call an oxidation reduction process. All right? So, what's happening is that these fuels, we'll call it fuels, glucose, right? That's going to be our ultimate input. Right? These carbohydrates or cellular fuel is going to be oxidized. All right? And we're going to talk about what oxidation reduction is in a second. Okay? There's basically two processes that occur for the oxidation of fuels. One is with oxygen, one is without oxygen. Okay? So fermentation is a partial degradation of sugars that does not require oxygen. Okay? It yields things such as lactic acid and ethanol, depending upon what the start is, the starting fuel is. All right, so yeast do this, bacteria do this, and they create things like beer and wine and whatever else. Okay? But it's without oxygen. In terms of the ultimate output of energy, fermentation is relatively low. All right, it does not produce a lot of ATP. Aerobic respiration, which requires oxygen, okay, consumes your organic molecules such as glucose, right, fructose, other organic other carbohydrate fuels, right? But it requires oxygen, and it yields a large amount of ATP, okay? Aerobic requires oxygen. There's a third thing in here, okay? Anaerobic respiration. Anaerobic means it doesn't require oxygen, but the respiration process occurs the same, but instead of utilizing oxygen at the end, it utilizes some other compound. Sulfur is one, nitrogen can be another one, right? And it can produce more ATP than fermentation, but usually less than aerobic respiration, okay? So there's 3 different catabolic processes. Eukaryotic cells will do fermentation, and they will also do aerobic respiration. All right, they really don't do the anaerobic respiration. Okay? But cellular respiration as a process, okay, includes both aerobic and anaerobic, but it's often just the aerobic respiration that we're talking about. Okay? The ultimate or the overall equation for this, for cellular respiration, C6H12O6 plus, oh no, it's broken down in the presence of oxygen. So let's erase this. All right, so plus 6O2, right, yields 6CO2 plus 6H2O plus ATP and heat. Okay? Roughly between 30 and 34 ATP in this process. Okay? That's the overall. Are you going to be asked to balance the equation? No, so don't worry about it. Glucose plus oxygen, because it's required, breaks glucose down. Cellulation breaks this down because it's a catabolic process, and energy is going to be released. You're going to return to this environment CO2 and water, and we're going to store ATP and release heat in the environment as well. All right, that's the overall process. Okay? Now, it does not mean that glucose is the only thing that can be utilized in this. You can utilize proteins, fats, other carbohydrates, okay? But the most efficient substrate to begin is glucose, okay? So that's the best bang for your buck, if you will, right, to start with. This process is a coupled or depends upon oxidation reduction reactions. All right? Oxidation reduction reactions are a transfer of electrons from one molecule to another. When an electron is transferred from one molecule to another, all right, that is an oxidation reduction reaction. When I say electrons are transferred, I want you to follow the hydrogens. All right, what did I say? I want you to what? Follow the hydrogens. Write that down in your notebook. All right, follow the hydrogens. Okay? Because really the hydrogens are going to tell you what molecule is going to be oxidized and what molecules are going to be reduced. Okay? So if we wrote our equation again, C6H12O6 plus O2 yields CO2, water, and ATP. Okay? I said follow what? The hydrogens. Where are the hydrogens going? Right? Yes? They left glucose and they went to where? They went to water. Right? They went to water. Okay? The molecule that is going to receive the hydrogens, receive electrons, is going to be reduced. So oxygen is reduced because it's going to gain electrons, hydrogens, and become water. Okay? Glucose is going to be oxidized. It's going to lose electrons, hydrogens, and become CO2. See what happened there? I took the hydrogens away, CO2's left. I add the hydrogens here, H2O is produced. Okay? So oxidized, right, there's where the hydrogens go. So glucose is going to be oxidized to CO2. Excuse me, is going to be reduced to CO2. No, I was right. Oxidize is losing. Oil rig. Oil rig. Oxidize is losing. Reduction is gaining. Got it? It's a little thing that I learned way back when. Okay? So, what is being oxidized is losing. Glucose is losing electrons in the form of hydrogen to CO2. Oxygen is being reduced. Reduced is gaining electrons in the form of water, in the form of hydrogens to form water. These are oxidation-reduction reactions. Oxidation loses electrons, or it's oxidized. Reduction is gaining electrons, all right, or is reduced. In this case, the amount of positive charge is reduced because you're gaining what? An electron, which is how charged, Nancy? Negative. All right, so it's a reduction of the overall positive charge. So what is oxidation is what? Losing. And reduction is? Gain. Okay? Why do we call these why do we keep them together? Oxidation, reduction. If something's being oxidized, it's going to lose what? Where does electrons need to go? Where? They have to go somewhere, right? Yes? They don't just dissipate into space. Okay? So, if something's being oxidized, something else has to be equally reduced because those electrons need to go somewhere. So we call them redox reactions, reduction, oxidation, because they're linked. Those electrons have to go somewhere. Okay, here's it. Same things in the case of sodium chloride, right? Movement of electron. Sodium ion is being oxidized, it loses the electron, and Cl, chlorine, is being reduced. All right. Now this is more vocabulary. I don't care, don't write this down. So you have a reducing agent and an oxidizing agent. That tends to confuse you a little bit because, right, the electron acceptor is the oxidizing reagent, right? If you know what's being oxidized and what's being reduced, that's good enough for me. All right, I don't need you to know the vocabulary too much. All right, here's another example. Reactants become oxidized to CO2, right? O2 becomes reduced to water. What do you see there? What are you following again? Following what? Hydrogens. Okay? Hydrogens are leaving and they're going there. Okay? The hydrogens end up right there. Right? Becomes reduced. Why am I we're really following electrons, right? So why am I telling you follow hydrogens? Because hydrogen has one electron, you have to follow, it has to go somewhere, right? It's not just, we can't just follow the electron by itself, and where the electrons go, hydrogen follows. Okay? So, what you end up doing, well, in these redox reactions, electrons go someplace. And electrons go to those atoms that have high electronegativity. Do you recall what electronegativity was? Mike just went, oh man, that was a month ago. What's electronegativity?
Ability for atoms to bind together.
Ability of atoms to bind together. Not really. Addie.
Electronegativity is like how likely they are to accept electrons from their valence.
How much they attract electrons to them. Okay, good. All right, so if you have a high electronegative atom, it's going to attract electrons, right? Oxygen is one of the highest electronegative atoms. So what's it do? It attracts electrons. Just like when we talked about the shape of the water molecule, its polarity is because oxygen has a high electronegativity. Those electrons are attracted to it, so you get polarity. That happens. Okay? So, the process of cellular respiration depends upon these redox reactions. The reduction and oxidation of certain molecules. Okay? I just gave you the overall equation. C6H12O6 plus oxygen yields CO2 plus H2O plus ATP. Right? And we just already said that glucose is going to be oxidized and oxygen is going to be reduced. That's the overall equation. Now, it doesn't happen like that in just one step. There is multiple steps along the way. And along the way, those electrons are going to be moved from glucose eventually to oxygen. And as that happens, all right, it's like a waterfall kind of, or a staircase. Every step you go down, you lose a little bit of energy, right? A little bit of energy is released. And as you step from one level to the next, down, energy is released, and the cell stores that energy in the form of ATP. Right? So doing it in a stepwise process allows the cell to efficiently produce ATP from glucose. Okay? So hydrogen, with its electron, is transferred to oxygen eventually. So we follow the hydrogens, you figure out where the electrons are going. Okay? There you go. There's your overall process. C6H12O6 plus 6O2 yields 6 CO2 plus 6 waters and energy. Roughly 30 to 34 ATP total from one molecule of glucose. Keon.
I just don't understand like how you follow like the arrows. Like why is it going to glucose to carbon dioxide? Why couldn't it go to water or something?
The carbons?
If we're following the hydrogens.
To follow the hydrogens, right? That's leaving glucose. So if I take the hydrogens out, what do I have left over? carbon and
Oxygen.
oxygen, which becomes this, okay? And now those hydrogens go out over here, happy, where are they gonna go?
The water.
They're gonna go to O2,
Yeah.
right, which then becomes water,
Okay.
okay?
Yeah.
Is that better?
Yeah.
All right. Now, I said this was a stepwise process, all right? And it requires an electron carrier. Okay, so because we're going to move the electrons off of glucose in a ordered fashion, we're going to pull them off, one by one by one. And then we're going to transport them, we're going to hold on to them. And we don't want to just dump them on oxygen right away, because then we can't harvest as much energy from them as necessary, all right, or as required. So there's a carrier that's there, NAD+, okay, which is a nicotinamide derived molecule. It is an electron carrier. It picks up these electrons as they're pulled off of glucose and holds on to them and transports them to the electron transport chain in the mitochondria, which allows for the ATP to be produced. Okay? So NAD is the electron carrier. NAD. And it is reduced to what? NADH. We're following the hydrogens. Okay? So NADH is the reduced form of NAD. Okay? And what it does, it says it represents stored energy. It has stored energy in it in the form of an electron that it will be transferred to the mitochondria and will be used to synthesize ATP, all right, in a very coordinated process. Okay? Questions on oxidation reduction? All right, here's nicotinamide in its oxidized form, okay, which means it's in the NAD form right here. Here's your two hydrogens, your two electrons are going to be added, and then you're going to get nicotinamide in the reduced form. We're really just looking at this part to that part, right? We're just adding the hydrogen right there. Okay? Questions on that? All right, but it is going to transfer that electron to the electron transport chain to produce ATP. All right, we'll